Thank you, and welcome to Synthego's webinar, Troubleshooting ICE, Guide Sequence Not Found. My name is Mike, and I am a technical support scientist at Synthego. In this webinar, we will discuss a common error message where ICE cannot detect the guide sequence in the control sequence. Here is what we will cover. First, we will talk about the limitations of ICE. Next, we will take a look at what the ICE error message for guide sequence not found looks like. Lastly, the ways to troubleshoot your sequence not being found will be discussed. Remember that you can download a PDF copy of the full slide deck to follow along and take notes during this webinar adventure. Let's get started. ICE can analyze a variety of SP-Cas9 mediated edits such as knockouts, small knock-ins, SNV, or codon swaps. However, it does have some limitations. When you are trying to knock out a gene using a single guide RNA, the largest deletion ICE can detect is up to 40 base pairs. When using multiple guides for your knockout, ICE can analyze deletions up to 150 base pairs. It should also be noted that 3 guides are the most that can be input into ICE for your analysis. For knock-in experiments, ICE has the capability to detect insertions up to 270 base pairs. ICE also assumes you are using SP-Cas9 nuclease and will place the cut site 3 bases upstream from the 3' end of the guide. Lastly, Sanger sequencing always has some inherent noise. Because of this, ICE cannot clearly determine if sequences present at 5% or less are background noise or if they are truly present. Even if these sequences are real, the sensitivity of Sanger sequencing is very low in this range. We therefore recommend cautiously evaluating any sequences that are present at 5% or less. Here you can see one common ICE error message stating the guide not found in control sequence. You should also note the ICE status says succeeded in orange, indicating that there was some parameter ICE was having difficult time interpreting from the uploaded information it received. In this instance, ICE gives you advice on how to correct the troubling results by stating the guide is not found in the control sequence. It is considered to check the guide sequence that was input into ICE and to exclude the PAM sequence. There are a few possible causes of ICE not being able to find your guide sequence in the control, and here are ways Synthego recommends correcting these issues. The first possible cause is that the guide sequence you initially entered into ICE is incorrect. Make sure the guide RNA sequence that was delivered into the cell is entered in ICE correctly and the PAM sequence is not included. PAM should never be included in the guide sequence since it is the region where the nuclease binds to DNA. A second possible cause is that the wrong section of the genome was amplified and sequenced. You should always verify the guide RNA sequence is present in the control.ab1 file in your favorite genetic software by copy and pasting the guide sequence into the wild-type sequencing file. You should see they both overlap at the target gene. The last common possible cause is a mutation in the guide RNA target region. This is caused by natural variations such as single nucleotide variants referred to as SNVs or single nucleotide polymorphisms referred to as SNPs are very common. Therefore, you need to align your wild-type sequence with the reference genome to identify any possible natural variants in the targeted region. If there are no SNV or SNPs present, your wild type and reference genome should align. If there are SNV or SNPs present in your wild type sequence, you will need to consider changing your guide design to include these variants. If you would like additional resources advice, how to analyze results, and other webinars, feel free to glance over all of these links to help you further. All of our resources are freely available on our website and are great tools to help you get started with your experiments. Here at Synthego, we like to help you with your CRISPR needs anytime. Please feel free to reach out to us and we will be happy to assist you. I would like to thank you for your time, and please visit our Help Center and website at www.synthego.com for more information.